All right, students, let's begin. Please switch on your videos. Start this class. Now, today we're going to be talking about the search for genetic material, because initially no one knew how actually the genetic material was there, how it was found, what was its nature, and everything related to that aspect. So today we're going to see how DNA was finally proved to be our ideal genetic material and how it was actually the DNA only. People at that point of time, they thought that it was DNA or it could be RNA or it could even be protein. So they did not know what it was actually. So that is why they started this search for the genetic material so as to know its biochemical nature. Now, let us move forward and talk about the different search. So the search, it began in the year 1926. And the first experiment that was done, it was the transforming principle. Now, this experiment, it was performed by Frederick Griffith. Okay, now what he did in the year 1928, that he started to work with Streptococcus pneumoniae. It was a bacteria that causes pneumonia. Okay, and he saw that how this Streptococcus particular bacteria, it changed, or what kind of transformation he observed. So see, this Streptococcus pneumoniae, it is also called as pneumonococcus, and this is a bacteria that has two types of colonies. Do you people know that bacteria, they have colonies? See, once the bacteria, like for example, if you've taken one bacteria and you are growing it in a petri dish, okay? After a while, after you can say 24 hours or even after 20 minutes, you'll see their population to be increased. So the increase or the population of the bacteria are called as the colonies. Okay. Now the bacteria Streptococcus, it produces two types of colonies. One is the like when you see the colony growth on a petri plate, you will either see that it is, like you can clearly see it's round and perfect color and shine. But sometimes it can be rough, it can be not that perfect color and all of that sort. So the pneumonococcus, it produces two types of colonies. One is the smooth colony. Okay, that gives a very smooth appearance once it grows. And the other is the rough colony. Okay, so he said that there are two strains, they produce two types of colony. So one is a smooth and one is the rough. Now this smooth and rough, if you people remember class 11 cell chapter, therein you people might have studied about the slime layer, the glycocalyx. You remember? Cell wall, slime layer, glycocalyx, which was like the extra protective layer. So it was like, it gave the bacteria sort of like a very glistening or shiny type of a surface. Now, those, like amongst the colonies, the S strain that was there, strain means the type, like type 1, type 2, if I say, instead of saying type, I'm saying strain. So strain, the first strain is the S strain, and it is smooth and shiny. Why? Because it has a mucus coating on its surface, which gives it a very shiny appearance. This mucus coat is made up of the polysaccharide. Now, this S strain is virulent. What is the meaning of virulent? It is like virulent means the organism will be causing disease, that is pneumonia, and it will make the host die. So S strain, all the S strain members, they were virulent. Whereas the R strain, it was rough, it has no mucus coating, and it is non-virulent. Okay. Non-virulent means it will not cause any disease. Now, what Griffith did is that he took all of these bacterial strains and then he started to inject it into the mice, and he wanted to see whether the mice was living or whether it died. Okay, so when the S strain, which is the virulent strain, when this was injected into the mice, obviously the mice got pneumonia and it died. When Griffith injected the R strain into the mice did not get the pneumonia and then he lived. Now, the next thing Griffith did is that he started to heat kill the bacteria. Okay, And when he gave the heat-killed S strain, meaning the strain that had now the killed bacteria, and when he injected that into the mice lived. Even though it was the S strain, but since it was a heat-killed S strain, so the mice lived. But then what he did is that he took the heat-killed S strain, combined it with the R strain. Now you know that R strain doesn't cause any disease, right? He took the living R strain and he combined it with the heat-killed S strain, and then he injected into the mice. And to his surprise, the mice died. Even though the S strain was killed, and only the R strain was there, which doesn't kill the mice, but still the mice died. So what is the case? So he said that maybe something from the S strain came to the R strain and transformed it to become a virulent type. This is what he meant by transforming principle. And when he actually saw the dead mice and did the mice's culture, he saw that S bacteria was found instead of R. So the conclusion that he came along was the R strain bacteria, it had somehow been transformed by the heat-killed S strain bacteria. And he said there is some sort of transforming principle that is changing the non-virulent into virulent. Okay, and he did not know what it was, but he said that maybe this is because of the genetic material. He still did not know what the genetic material actually was. But he sort of made a speculation that, okay, this transforming material is indeed a genetic material. So is this transforming principle clear to you all? Did people get the whole concept behind it?
Yes,
Everyone got it?
yes, ma'am.
Okay, what about the others?
Yes, ma'am.
Okay.
Yes, ma'am.
All right, now let us see the biochemical characterization. Now, people, once the transforming principle was out, then people started to search more on it as to find out what is actually happening. So, now since Griffith said that it was the genetic material, now people wanted to find out what is the nature of this genetic material. So, earlier, people thought that genetic material was a protein, but later on, let us see. So in the year, like from 1933 to 1944, these scientists that were Oswald Avery, Colin MacLeod, and Macklin McCarthy, they purified the different biochemicals such as protein, DNA, and RNA from the heat-killed S strain, meaning they took the S strain, they heat-killed it, and they took out the protein of that bacteria, they took out the DNA of that bacteria and RNA. And later on, they found out that it was the DNA that was causing the transformation. Now how? It's because when along with that combination of S strain and R strain, when they added protease are the enzymes that degrade the protein. RNAs are the enzymes that degrade the RNA. So when they added these two enzymes, still the transformation took place. Nothing happened to the transformation. So with this, they thought that, okay, it could not be a protein because even if the protein was degraded, still the transformation was happening. Then they thought, okay, it couldn't be an RNA because when the RNA was degraded, still the transformation was happening. Later on, when they added DNA's enzyme, the DNA degrading enzyme, and then they saw that the transformation stopped. So ultimately, it was concluded that DNA was indeed the genetic material. Did you people get this biochemical characterization? Any doubts? Okay. Now let us see. Now people, once this was speculated, then people started to find the proof that yes, indeed, DNA was the genetic material. So there were two people, Alfred Hershey and Martha Chase, in the year 1952. They started to work on bacteriophage. Now, what are bacteriophages? The bacteriophages, these are those kind of viruses that infect the bacteria. You know that There are viruses that infect the humans, that are viruses that infect the plants, and also the bacteria are not left behind. So there are viruses that even infect the bacteria, and the bacteria infecting viruses are called as bacteriophage. And this is how they look like. You might have studied in class 11 if you remember biological class. So the bacteriophages, what it does is that, for example, if this is a bacterial cell, it's not this big, but we are enlarging it. And the bacteriophage is sort of like this. So it sits on the bacteria and it has a genetic material. So what it does is that it releases its genetic material into the bacterial cytoplasm. Okay, this is what bacteriophages do. Why? So that the bacteria, when it divides, it could make more bacteriophage. So bacteriophage, they attach to the bacteria and enter its genetic material into the bacterial cell, and it makes the bacteria to manufacture more and more viral particles instead of bacterial particles. So now Hershey and Chase, they were working in order to find out whether it was the protein that the bacteriophage was giving to the bacteria or whether it was the DNA that was being sent. So what they did is that they grew the virus that contained radioactive phosphorus and the others that contained radioactive sulfur. So they took two petri dishes. Okay, one like he took like one of the viruses, and then they started to grow in a medium that contains radioactive phosphorus. Radioactive elements, you people understand? You might have studied in chemistry. Radioactive elements or that are there. So what they did is that they took the virus and they started to grow it on the radioactive phosphorus. Second medium, they took radioactive sulfur and he grew the viruses on that. So two media they were using and the viruses were grown on both of them. Now, you people answer me. What are the components of DNA? What all biomolecules do you find in DNA? The question is to everyone. Also, everyone, please switch on your videos and you're supposed to keep it on throughout the class. Yes, so what are the different biomolecules that you find in a DNA? Tell me, who can answer? You can try to remember the elemental stage of the DNA. What are all the things that you find in the DNA?
Nucleic acids.
Yeah, nucleic acid was there. Nucleic acid is like DNA and RNA are nucleic acids. So you have to tell me what are all the things that you find in a nucleic acid. That all of you know this. I've taught all of you. So now you tell me.
Proteins, amino
Yes.
acids.
Yes, proteins are there. Then proteins are made up of amino acid. So apart from that, also Aisha, please switch on your video. What else? What else do you find, students, in a nucleic acid?
Nitrogen bases.
Yes, nitrogenous bases are there. Correct. What else?
Phosphate groups.
Phosphate groups are there. Then
Deoxyribose sugar.
sugar is there, right? And sugar contains carbons and hydrogens and oxygens, right? Now, how many of you know what proteins are made up of? Just said it. Amino acids, right? So when multiple amino acids come together, they make the protein. And there are many amino acids that contain sulfur in. Okay. Now, the whole concept behind this is that, for example, DNA has phosphate group present in it. And if you are growing the virus in the radioactive phosphorus, so this radioactive phosphorus is going to get linked with the phosphorus of the DNA. Okay. And imagine it like I used to imagine it. I used to imagine it like, imagine the phosphorus shining. If it is shining, then that means it is radioactive. Clear? This much you've understood? Does anyone have any doubts? Okay. If wherever you have doubts, please feel free to ask, because this is a very you can say tricky, and it's like not related to whatever you people have studied up until now. So wherever you want me to explain things again, please do, because the questions get asked a lot from these topics. Apart from that, now that protein, it contains amino acid, and many amino acids are sulfur containing amino acids. So this sulfur will get connected with the protein part. Okay. So now you have labeled, or you have tagged the DNA and the protein. Now, the virus that is growing in radioactive phosphorus medium, it will contain a radioactive DNA, because this phosphorus got incorporated into the phosphate of DNA. Clear? But this virus will not have a radioactive protein, because protein has no phosphorus place to get it, right? But if, because DNA contains phosphate and the protein does not. Now, the virus that grows on the radioactive sulfur medium, then it will have the radioactive protein, but it will not have radioactive DNA. Everyone's following me till here?
Yes,
Now
ma'am.
see, during this whole entire procedure, they took 3 steps of the experiment: infection, blending, and centrifugation. Now, they, like these radioactively labeled bacteria, one was the phosphorus, the other was sulfur. They took it and they allowed it to infect the bacteria. This was infection step. Then the contact between them was broken. Now they thought that, okay, since it has now infected the bacteria in this amount of time, now whatever it was, the genetic material, that had been transferred. Now they caused the blending. Blending means now they remove them from each other. Lastly, centrifugation means the viral particles, they were separated from the bacteria by spinning them in a centrifuge. Okay, so the heavier one settles at the bottom, lighter one remains at the top during centrifugation. Now, after the whole entire centrifugation was done, then the bacterial cells, once they were shown, or you can say they were studied, now they wanted to see that whether in the bacterial cells will we find radioactive sulfur or will we find radioactive phosphorus. And at the end, the radioactive phosphorus or the radioactive DNA was found in the medium. This proved that, okay, DNA is indeed a genetic material. Is it clear? Did you people understand this experiment performed by Hershey and Chase?
Yes, ma'am.
Everyone understood it? Crystal clear?
Yes, ma'am.
Okay, very good. Now, once you people will take down the notes, you'll understand it much better. But this was the experiment that was done. Now, let us see. You people remember when I was teaching you the reproductive health chapter, in that I taught you what are the properties of an ideal contraceptive. Same way, we are going to see the properties of the ideal genetic material. First of all, whichever genetic material, it could be anything, but it should be chemically and structurally stable. Okay, whenever you people write, remember that I am writing chemically and structurally. Don't write struct and chem, because your teacher will not understand. I will, but your teacher will. So chem, no, chemically and structurally stable. So it should be stable, stable as in, because it gets transmitted from one generation to another, so it should not change its chemical structure, or it should not change the way it looks. First point is that. Second, it should be able to make its copies, right? It should be able to make more and more copies of itself. It should have the scope of slow mutation. See, slow mutation is very much welcome, but fast mutations are avoided, because fast mutations is equal to disaster. Slow mutation is equal to new characters coming. And lastly, it should be able to express itself in the form of Mendelian characters, right? It should be able to get inherited. It should be, like we should be able to understand that, like the chromosomal theory and everything, it should align with that. And that is what, like, there are the different, you can say different properties of an ideal genetic material. Now comes along why only the DNA is a preferred genetic material and how and why cannot the RNA be our genetic material? There are many reasons, some of which are because you people know that RNA, it has an extra OH group, which is at the second position of the uh, pentose sugar. Now that OH group is free. So it can react with anyone. It can react and form condensation reaction, different reactions it can do. So which makes the RNA labile, which is like easily altered and easily degradable. Secondly, you know that RNA has uracil. It doesn't have thymine. But thymine gives more stability as compared to the uracil. So presence of thymine instead of uracil, it gives DNA more stability. So you could write it in your own language also like this, that since RNA has uracil instead of thymine, it is less stable than DNA, like that. And lastly, RNA mutates very, very quick because it is unstable and we cannot afford that. So because of all of these reasons, RNA is not taken as a genetic material. But you people will be surprised to know that it was RNA only that was first ever to exist on the earth in the form of genetic material. It was not DNA. Later on, the DNA evolved. So RNA was the first genetic material, as many life processes such as metabolism, translation, everything evolved around the RNA. And RNA, it used to act like a genetic material, but since RNA is so reactive, it acts like a catalyst. What is a catalyst, students? A catalyst.
It increases the speed of reaction.
Yes. Right? It like increases the rate of the reaction, it lowers the activation energy. So RNA is a catalyst. But DNA, it evolved from RNA. Okay. So DNA now, like DNA had double-stranded, standard makes strands. DNA is made up of two strands, and the strands, they are complementary to each other. Because of these two very reasons, they resist any change, meaning it's not very easy that the DNA could get changes in it as such, okay, as compared to the RNA. So now you people have understood the whole entire thing. Any doubts? Okay. All right, now after that, we're going to see how the replication of DNA happens. But before this, you people note down till here, and then we'll move forward. Yeah, now you can switch off your video, students. You can switch off and write down comfortably. Everyone's done till here?
Yes, ma'am. What's before the saw miraculous transformation?
That's and. And.
Ma'am,
Yes, dear.
what's cultural plate?
Culture plate. See, whenever you want to grow anything, okay, whenever you want to grow anything, you like, for example, you're growing bacteria, you're growing viruses. So you require a media, okay? Like you can't grow it just like that. So a media is required, and culture plate, it's like a petri dish which contains the media for the bacteria or anything of that sort to grow. So that is what the culture plate is. Okay, it's like a petri plate that has the media so that this thing, the bacteria or the virus, anything that could grow. Is it clear?
Yes, ma'am.
Are you people able to see the screen?
Yes, ma'am.
Ma'am.
Oh. Is everyone done till here? Shall I scroll down?
And the screen is not visible.
It's not visible. Okay, this one.
No.
Is it visible?
Yes, ma'am.
Okay. Shall I scroll down?
1 second. Can you scroll up? I wasn't able to see the screen before.
Okay, okay. Now is it visible to you?
Yes, ma'am.
Okay.
Done, ma'am.
Ma'am, what is the meaning of strain?
Yeah, strain means it's like a you can say, imagine it like type. How we say this type of bacteria, that type of bacteria, or this type of colony, that type of colony. So the types of colony is what we call as the strain. Imagine strain means type. Like this is our S strain is first type of strain, and then R strain is the next type of strain.
Ma'am, is pneumonia the S strain?
No, serious. S strain is the type of bacteria that causes pneumonia. Okay, S strain is a virulent strain. Virulent means infective strain. So if the mice gets this particular strain in it, that mice will get pneumonia and the mice will die. But if we are talking about the R strain is called as the rough strain, and it has no mucus coat and it's non-virulent. Non-virulent means it cannot cause the pneumonia. So if we inject the R strain into the mice will live. Okay.
So if this S strain bacteria is like present, it will cause pneumonia?
Yes, it will.
Okay. I'm done.
Everyone's done till here?
Yes, ma'am.
What's heat killed?
See, also one more thing, students, please try to join the class on time. Just because like you people miss if you don't join on time. See, Aisha, what he did in his experiment is that he started to inject the different types of bacteria into the mice, and he wanted to see whether the mice is going to live or whether the mice is going to die. So when he put the S strain, normally the mice died because the mice got pneumonia. When he injected the R strain, that was like non-virulent, so the mice did not die and it lived. And then he heat killed the bacteria, meaning the bacteria, he killed it. And then he took that killed bacteria and he injected into the mice. Now, since the bacteria was killed, the mice did not get any pneumonia from the S strain and the mice lived. But then he made a combination of the heat killed S strain bacteria and the R strain that was live. And then he injected into the mice and the mice died. Because originally the mice shouldn't have died because the S strain was heat killed and the R strain doesn't cause pneumonia. So he said that, okay, there was something that from the S strain, it was being sent to the R strain and it was making the R strain become virulent like S strain. Later on, S strain was recovered from the dead mice and it was finally, you can say, they said that this is because of the genetic material that is getting transformed, the other type of bacteria. Okay, he just speculated that this is because of genetic material, but up until his research, people did not know what is going to be the genetic material's nature, whether it is protein, whether it is RNA or DNA. Is it clear?
Yes, ma'am. I'm done.
Everyone's done till here? Done, student?
Yes, ma'am.
Students, are you done? Is everyone done till here? Yes or no?
Yes, ma'am.
Also, once you people are done, please unmute and speak. Okay.
Ma'am,
Yes, dear.
what's degrading? Like, what do
Degrading, example, if degrading means something that destroys the other thing. For example, proteins are there, and if you apply the protease enzyme, okay, protease. Now, this protease enzyme, it is going to degrade the protein and make it, like, degraded means to destroy it. Same way, if RNA is there, then you should apply RNAs. See, the any word that is followed by ase as the suffix, it means degrading. Okay, so RNAs, this is an enzyme that will degrade the RNA. Same way, the DNA will be degraded by the enzyme called as DNase. Okay, so he applied all of these enzymes and to see whether which of the material got degraded and which was being sent, like that. Clear?
Yes, ma'am.
Yes, ma'am.
All of you are done, students?
Yes.
Okay. Done, students? All of you are done till here?
No, ma'am.
Shall I scroll? Everyone's done?
Yes, ma'am.
Done, students?
Yes, ma'am.